Hello everyone. Today I will demonstrate the ELISA technique, which stands for enzyme-linked immunosorbent assay. This method is widely used to detect and quantify proteins, antibodies, and biomarkers in biological samples.
The technique is based on a highly specific interaction between an antigen and an antibody. When the target molecule is present in the sample, it binds to a specific antibody attached to the wells of an ELISA plate. After a series of incubation and washing steps, an enzyme-linked detection system is added. Then the enzyme reacts with a substrate, producing a color. The intensity of the color is directly proportional to the amount of the target protein present in the sample. Before opening the ELISA kit, let's first examine the information printed on the kit box. We can see the manufacturer's name, the assay parameter, which in this case is rat brain-derived neurotrophic factor, so the kit is specific to rat samples. Next, we have the catalog number. Then, the kit size, which may be 48 wells or 96 wells. Here, we can find the manufacturing date and the expiration date. Note that most of kits are valid for 6 months or 1 year. Another important detail is the storage condition. Here, the kit should be stored at 2 to 8 degrees Celsius, so it should be stored in the refrigerator, not the freezer, and the storage instructions must be adhered to in order to ensure the validity of the kit. Before we start performing the ELISA assay, let's take a look at the contents of our ELISA kit and understand the purpose of each component. Inside the kit box, we can find the following items. First, we have the instruction manual or pamphlet, which contains the assay protocol, including reagent preparation, incubation times, and calculation methods. It is important to read it carefully before beginning the experiment. Next, we have the ELISA microplate, which contains 48 or 96 wells which are pre-coated with the primary antibody against the target antigen, so this is where the standards and samples will be added, and where the antigen-antibody reactions will take place. We also have the plate sealer. After adding samples or reagents, the plate is covered with this sealer to prevent evaporation and contamination during incubation. Note that we can use PAR film instead of the provided plate sealer. Also, we have the standard, which contains a known concentration of the target protein, and the standard diluent, which is used for preparing serial dilutions of the standard solution to get the standard curve. Next is the wash buffer concentrate, which must be diluted according to the kit instructions and used to wash the plate to remove unbound substances. We also have the biotinylated antibody, which binds to our target protein. After that, we have the HRP conjugated antibody. This reagent contains the enzyme horseradish peroxidase, which is responsible for generating the color upon reacting with solution A and B, which are the substrates. They react with the horseradish peroxidase to produce a blue color. Finally, we have the stop solution, which stops the enzymatic reaction and changes the color from blue to yellow, allowing accurate measurement of the optical density. Now that we are familiar with all the kit components, we are ready to start the ELISA procedure step by step. First, let's identify the wells on the ELISA plate. The plate is arranged in rows and columns. The rows are labeled with letters A, B, C, D, and so on, whereas the columns are numbered 1, 2, 3, and so on. This labeling system helps us accurately locate and record the positions of standards and samples. To avoid confusion while working, I place the ELISA plate on a white paper with a black dot, and the well currently being used is positioned directly over this dot. This simple technique helps me keep track of my position on the plate, so I can identify the correct well and reduce the risk of adding reagents to the wrong well. To make it easier to keep track of the wells during the assay, I use a color coding system. Each row is assigned a different color. For example, row A is marked in red, row B in yellow, row C in orange, row D in blue, and so on. Then, I record a color-coded key in my notebook. For instance, A in red represents the standards, B in yellow represents the control samples, C in orange represents the model samples, and D in blue represents the treatment samples. This helps me stay organized and accurately identify each row during the ELISA procedure. Of course, before starting the assay, I carefully review the pamphlet and prepare a simplified step-by-step summary in my notebook. I organize all the instructions in a clear and easy-to-follow format so that I don't get confused or miss any step during the procedure. The first step is to prepare the serial dilutions of the standard. Let's say the provided standard has a concentration of 80 mg per milliliter. We will prepare serial dilutions to obtain the following standard concentrations: 40, 20, 10, 5, 2.5, and 1.25 mg per milliliter. These serial dilutions will be used to generate the standard curve. First, I prepare a set of tubes and label each tube with the concentration that will be prepared, like 40, 20, 10, 5, 2.5, and 1.25. Next, I use the provided standard diluent supplied in the ELISA kit to prepare the serial dilutions of the standard. According to the pamphlet, the first step is to add 120 microliters of the standard diluent into each labeled tube. Next, I will transfer 120 microliter of the stock standard into the first tube labeled 40. Then, I place the tube on the vortex mixer and mix it thoroughly. Next, I transfer 120 microliter from the tube labeled 40 into the tube labeled 20. Then I mix this tube thoroughly using the vortex mixer. After that, I transfer 120 microliter from this tube to the next tube labeled 10 and repeat the same process till the serial dilutions are ready. Now, let's start loading the ELISA plate. And to ensure that I am working with the correct well, I first align the designated well with the black dot on the white paper. According to the protocol in the pamphlet, I will add 50 microliter of each standard into its designated well. So, I carefully add 50 microliter of the stock standard provided with the kit with concentration of 80 mg per milliliter into the first standard well. Then I add 50 microliter of the diluted standard with concentration of 40 mg per milliliter into the second well. Next I add 50 microliter of the diluted standard with concentration of 20 mg per milliliter into the third well. And so on. Afterwards, according to the protocol, I will add 50 microliter of HRP conjugated antibody to each standard well. Next, we will load the sample wells. According to the protocol, I will add 40 microliters of each sample into its designated well, followed by 10 microliters of the biotinylated antibody and then 50 microliters of the HRP conjugated antibody. Please note that the micropipette tip should never touch the well. Instead, dispense the reagent carefully into the center of the well without allowing the tip to come into contact with the sides or the bottom of the well. Also, make sure that the well you are currently working on is always aligned with the black dot on the white paper. Next, I seal the plate with the provided sealing film or PAR film. Then I place it on a shaker with a white sheet of paper underneath. I always start the shaker at the lowest speed, gradually increase the speed for gentle mixing, and then gradually decrease it back to the lowest speed before switching the shaker off. Next, I place the sealed plate inside the plastic oven or incubator on a white sheet of paper. The plate is incubated at 37 degrees Celsius for 60 minutes. After the incubation period is complete, I carefully remove the plate sealer or PAR film. Then, I gently invert the ELISA plate over a stack of tissue paper placed on the bench to discard the contents of the wells. This step removes the liquid from all wells before proceeding to the washing step. Note that, during the plate inversion step, the strip wells may accidentally fall out. Thanks to the color coding system and the key recorded in my notebook, I can return each strip to its correct row. For example, the strip labeled in red goes back to the first row, the strip labeled in yellow goes back to the second row, and so on. This helps prevent any mix-up. Also, that's why I always discard the plate contents onto tissue paper on the bench. If any well strip accidentally falls out or becomes detached during this step, it will stay on the bench, allowing me to place it back in the correct position according to my color-coded key, rather than falling elsewhere and becoming mixed up or contaminated. Now, we need to prepare the washing buffer. The kit provides a 25x wash buffer concentrate, which means that for each 1 milliliter of the wash buffer concentrate, I will add 24 milliliters of distilled water to prepare 25 milliliters of diluted wash buffer. Note that we can dilute the washing buffer using distilled water, purified water, or water for injection. According to the protocol, I add 350 microliters of the diluted wash buffer to each well and let it stand for 1 minute. Then, I gently invert the plate and discard the contents onto tissue paper on the bench. This washing step is repeated a total of 5 times to ensure that all unbound materials are completely removed from the wells. The final step is to add 50 microliters of solution A and 50 microliters of solution B to each well. Please note that solution B is light sensitive and is therefore supplied in a brown bottle to protect it from light. For this reason, this step should be performed in the dark or under minimal light exposure to maintain the stability of the reagent. After adding solutions A and B, I seal the plate with par film. Then, I place the plate on the shaker with a white sheet of paper underneath. I start the shaker at the lowest speed, gradually increase the speed to ensure proper mixing, and finally reduce the speed back to the lowest setting before turning the shaker off. Next, I place the sealed plate inside the plastic oven or incubator on a white sheet of paper. Then, I incubate the plate at 37 degrees Celsius for 10 minutes. After incubation, a blue color develops in the wells as a result of the enzymatic reaction. Next, I add the stop solution to each well. The stop solution terminates the reaction, causing the color to change from blue to yellow. The intensity of the yellow color should be measured using an ELISA microplate reader within 10 minutes of adding the stop solution. Reading the plate within this time ensures accurate and reliable results. After placing the plate into the ELISA microplate reader, the software, such as Gen 5, asks which wavelength should be used to measure the optical density. According to the protocol, I select 450 nanometer as the measurement wavelength. The instrument then measures the optical density of each well and generates the results. Now we have the optical density values for all the standard wells. And since the concentrations of these standards are already known, we can use these data to construct the standard curve. This standard curve gives us an equation. By entering the optical density value of each sample into this equation, we can calculate the concentration of the target protein in this sample.